Good evening to all. I would like to invite Professor Gilles, Deputy Head of the Institute of Chemistry for Life and Health Sciences, PSL University, CNRS, France. And also I welcome Dr. Rajendran Venugopal, Assistant Professor, Central University of Tamil Nadu, India. Both, thank you for accepting our invitation. Thank you, sir. Now session is over to chairperson.
Good evening to everybody. It's my great pleasure to introduce Professor Gilles as the speaker of this session. He is Deputy Head of the Institute of Chemistry for Life and Health Sciences, PSL University, CNRS, France. He was a Swiss National Science Foundation Assistant Professor, 2011 to 2016, University of Zurich, Switzerland. He was a Swiss National Science Foundation Ambition Research Fellow, 2010 to 2011, University of Zurich, Switzerland. He has 5 years of postdoctoral research experience at Ruhr University, Bochum, Germany, and Monash University, Australia. He obtained his PhD from University of Neuchâtel, Switzerland. He has received several awards for his excellent scientific contribution in bioinorganic chemistry. If you read his awards he received, it takes more than 5 minutes to read all the awards he has received. I highlighted 1 or 2 important awards. Particularly, he received 2020 Churchill College Fellowship Award. In 2018, he received Aerobic Medal Award for work in bioinorganic chemistry. The year of 2016, he was recipient of Ernst Zucker Prize for carrying out research in the cancer field. And also in 2016, he was awarded the Journal of Medicinal Chemistry award. In 2015, he was a recipient of very prestigious Werner Prize Award for doing research or doing outstanding research in the field of chemical field. So this also he's author of 1 book, and he contributed 5 book chapters, and also he's published 27 reviews and 133 research articles. And also he has filed 11 patent applications, and he received 7 patents. His research contributions are much appreciated with citation 6,400 citations, and his publication has 38 H index. Currently, his research interest focusing on medicinal organometallic chemistry, anti-cancer and anti-parasitic drug development, inorganic chemical biology, phototherapy, and bioorganometallic chemistry. Everyone knows that he's a great scientist from his scientific contributions. In addition to scientific achievement, he is also equally good in sports. So most of us may not know that. So he was a snowboard instructor, qualified as a snowboard instructor. And also he was a hockey player, and he was a semi-finalist in the Swiss Cup 2003. And also now he qualified as floorball coach, hockey floorball coach. So in addition to scientific collaboration, those who are interested in sports, you can also contact him, have a sports collaboration. So he's such a multi-dimensional personality as all talents. We are proud to have Professor Gilles for today's session. Now, with your all permission, I invite Professor Gilles to tell you a talk on metal complexes in medicinal chemistry. Professor Gilles, please.
Okay. Can you hear me?
Yes.
Yes.
Yeah.
Cool. I will just share my screen now. Is it okay like this?
Yeah.
Hang on. If I'm changing, can you see?
Yes.
Is it the second slide?
Yeah, next, yes, move.
Okay, cool.
Go back to first.
Superb. Thanks a lot. So it is really a great pleasure to be with you today for this conference. And that is the first time that I'm doing a conference online, and I'm extremely happy to do it with two great colleagues here in India. Professor Rangazami was a postdoc in my laboratories in Zurich, and I met with Rajan Dhiran in Zurich a few years ago. Thanks a lot for the invitation. What I want to do today is to do something a bit different to that you have already heard during the conference, is to talk about metal complexes. What you can see on this slide is that all our students in our group are working with metal complexes, and they are working for medicinal application, different medicinal application. And what I want to do today is to show an overview of three topics: chemotherapy, phototherapy, and radioimaging. And I want really to show you the power of metal complexes in medicine. As you can see on this slide, and I hope you can see it, that I have changed slides. Let me know if it is not the case. Metal complexes are extremely useful in medicine. For example, if you take cisplatin, you will hear very, as a surprise for most of you, is that it is used in more than 50% of all chemotherapeutic treatment. It means every time someone is treated for cancer with a chemotherapeutic agent, a platinum compound will be given to the patient. The same is true, for example, with gadolinium. Gadolinium is heavily used when you are going for MRI. I have, for example, to go for an MRI every year to see my brain, and I'm receiving about 9 grams of this compound. And I like to highlight this 9 grams because it clearly shows that the common belief that metal complexes are toxic is wrong. It really depends about what is around the metal ion. Here in this case, this chelate is extremely stable, it stabilizes the gadolinium, and it is absolutely fine. The patient afterwards will urinate this compound. And we can keep going. Technetium-99m is used about in 85% of all imaging agents, for example, for heart. And also I have put 3 different examples here with arsenic, mercury, and gold. And this I have done intentionally because normally such metals you will believe will never be used in medicine. And it is not true. This is an anti-arthritic agent. You have probably already received a mercury compound because it is used as a preservative in vaccine. And RS-203 is now the frontline chemotherapeutic treatment in certain cases of leukemia. Why is that? Why do we use metal complexes in cancer therapy? It is because they have properties which are different than organic compound. It is complementary. It's not one against each other. It's just to say that they are complementary. If we can use an organic compound to make a treatment, let's use an organic compound. But sometimes metal complexes, because they have those properties which are unique, are very helpful. Ligand diction, just think about the mode of action of cisplatin. You inject the compound into the vein of the patient because it is, there is a high concentration of chloride in the blood, it stays stable, it can enter cells, but because there is a concentration which is about 100 times lower in cells than in the blood of chloride, you will have an exchange of the chloride with water. Finally, the compound will covalently bind to DNA. And this covalent binding is unique to metal complexes. Just think also about the redox activity. A metal ion can have different redox states. And this is very, very important, as you will see, for example, with ruthenium complexes. They have also a higher structural diversity than organic compound. If you take a ruthenium, you can put 6 different ligands around the ruthenium, and you can get up to 30 isomers. It's Eric Megers who was explaining this. On the contrary, if you take a carbon and you put 4 different ligands, you can only make 2 enantiomers. And for sure, the structural variety, the structural diversity that you can obtain with metal complex is much higher. Catalysis is also something which is quite unique for metal complexes. Radioisotopes are also extremely present with metal, but I have to acknowledge here that it is also the case for non-metal. The first topic that I want to discuss has been pioneered by Malay Patra, who was a postdoc in my labs in Zurich. He is currently an assistant professor at the Tata Institute of Fundamental Research in Mumbai. And this is a collaboration with my good friend, Professor Thomas Mint, who is now at the Medical University in Vienna. And it is about immunopositron emission tomography. What is this? First of all, I just have to make a little disclaimer that we own a patent and that we have given the right to use this patent to a company, as you will hear later during this talk, to ABX Advanced Biochemicals, which is located in Germany. So immunopet is a well-known imaging technique, for example, to detect tumors or metastases. What you can see here is a patient with lung cancer, which has already metastasized. What they have done here is to inject FDG. So it's a fluorinated glucose, which contains a fluorine 18. What you can do with this fluorine 18 is that it is a positron emitter. This positron can react with an electron to form 2 gamma rays. Those 2 gamma rays are ejected and can be detected by a gamma camera. And this way you can see where the compound is going. Because tumors are extremely prone to take up sugar, you can see that the compound is going mainly into the tumor as well as the metastasis. You can also see in the kidney because it's eliminated. This is already in the urine. And you can also see that there is a lot in the brain because, as you know, the brain is in high demand of sugar. But what you can see that the patient has really bad cancer. And after a treatment, not with radioactivity, but for example, with cisplatin, you see that the patient is in remission after 2 weeks and finally free of cancer after 6 weeks. This is fantastic and it is heavily used to detect cancer. The only trouble, for example, if you wanted to detect a brain cancer, you could not use this agent because there is already a background. That is why there are other isotopes like gallium, like copper or zirconium. What you can also see here, it's important information about the half-life of the isotope, how long it takes to have half of the activity released by the isotope. And you quickly see that gallium and fluorine are extremely quick, while copper and zirconium are much longer. And why is this very important? Is because if you wanted to use antibodies, which are really well known biomolecule to target certain types of cancer cells, you will not be able to use gallium or fluorine because an antibody takes extremely long to reach the tumor. It takes up to 3 days. That is why zirconium 89 is absolutely perfect for such application, for immunopet imaging. Immuno is to remind about antibody. For the moment, it is used in the clinic, and what is a bit surprising is that the ligand which is used is called DFO, desferoxamine. You can see that DFO complex the zirconium 89 or even the cold version, and through the hydroxamic acid. The only trouble is zirconium is octadentate. And because DFO is only hexadentate, you have 2 water molecules binding to zirconium, which is definitively not ideal because when you will inject this compound, you can see that you have an amine here to make a coupling, for example, to an antibody. the water molecule will be exchanged with something else oxophilic. And what is extremely oxophilic, for example, in the body are the bones. And what you don't want is to have an unspecific accumulation of radioactivity in the bone because there is a presence of bone marrow there. That is the reason why Malai was working on this project, and he prepared an octadentate version of DFO that we have called DFO star. Here you can see that there are not 3, but 4 hydroxamic acid, and not surprisingly, now the zirconium is able to completely complex DFO star, and it is now a happy zirconium 89 because its coordination sphere is fulfilled. What we could do is a synthesis which was extremely simple using cheap reagent because we want absolutely to push this to our clinical trial. Malai used 1, 2, 3, 4, 5 step synthesis to prepare this compound, which can afterwards be reacted with a commercially available DFO. You have now this compound here, you just have to deprotect it to have the DFO star version of the chelator. What we have done afterwards on the amine here is that you react this compound here in order to have an NCS handle, as you can see on the ligand here, DFO, which enable an easy coupling with antibodies, for example, with lysine present in antibodies. This was good. Now we had to prove that this compound, this chelator, was much better than DFO. That's why we have done transchelation experiment. We had a huge excess of a chelator and see if the complex which was formed is unstable. And you can directly see on this graph, in blue is DFO. You can see that there is a transchelation, why it is not the case with our chelator in red. We have then published this, and we receive an email from Daniel Wurks and Guus van Doggen in Amsterdam, and they say, look, it is very interesting, your paper, but you are a bit lucky because we were just about also to publish this compound, because indeed it is exactly what the nuclear imaging community is needed. And they say, look, we should combine force because we are working in a hospital. Send us the compound and we will test it on mice. And this is what they have done. We have sent them the compound here. They have attached it to trastuzumab, which is a well-known approved monoclonal antibody. And then they have labeled it with zirconium 89. I remind you here that the labeling must be done very fast and also in a temperature which is not destroying the antibody. And all of this was possible with our chelator. And very importantly, in terms of clinical application, we are using exactly the same condition than for DFO. So it means the nurses or the doctor in a hospital, we would have just to say, just use this bottle instead of the other one. What we can see afterwards during the in vivo experiment, you see actually on the left, 2 mice which have been, that's the same mice, but it's 2 different images. We have put 2 tumors on the side of the mice. It is a colon cancer. And with DFO star, you can quickly see that all the radioactivity is localized in the tumor. While for DFO, this is what is commercially available, what is typically given to patients. You see a lot of activity, which is on the spine, which is on the knee, which is on the leg, which is clearly meaning exactly what I have explained to you before. The DFO, which is not fulfills the coordination, it will enable the zirconium to bind to the bones, which is not what you want. What we could do afterwards, it's what they have done in Amsterdam. They have killed the mice and they have looked at the total amount of radioactivity in the bones. And you can see with DFO star, we have a much lower activity in bone compared to DFO, exactly as we were anticipating. We have done afterwards also a few videos. Here you can see the joint, you can see the tumor, the joint, and if we turn, hopefully we will see, oops, sorry, I wanted to start a video. I don't know why it's not working. Okay, that's not a problem. But you can really see that DFO here, you have a lot of unspecific radioactivity on the board, which is not the case at all with our compound, where you have only and only the activity in the tumor. What we were very happy is that the company in Germany has now commercialized this compound, so people can buy it to make more tests. And very interestingly, we wanted to keep going. Can we do something better? What we wanted, first of all, is maybe to solve a problem that is a bit, how to say, it is not ideal for the moment, DFO and DFO star, is because the compounds are not very well water soluble. It is fine to work with this compound, but if we could make a derivative which could be even more water soluble, it would be better. So what we had have a look, it's in the literature, and you can see different compounds which have been prepared by other people. And you can see that generally the yields to prepare the compound are relatively low. Okay, that's the first step. Then there is really this problem of water solubility. And very interesting, Rachel Codd in CIMI reported a few years ago a DFO star version which contained, as you can see, in the backbone, there is an ether. The X is the ether, which improves a lot the water solubility of the compound. However, to do this compound, it was requiring a lot of tedious synthesis, and she could only get a few milligrams, which was not enough to do radiochemistry. That is why we decided to use another approach to make such a compound, which is very good because it improves a lot the water solubility. It's to use solid phase synthesis, as it is currently done, for example, for peptides. The idea was to prepare oxo-DFO star here with the 4 extra oxygen atoms. But in order to do this, we wanted really to do this peptide coupling on the solid phase. Therefore, we have prepared here this symptom, which can afterwards be added on the solid phase to the resin. Manon was a PhD student from France, and she did this synthesis. Once again, you can see on this slide, I'm going a bit quickly on this slide, but it is very common synthesis. There is nothing unexpected, and the yields for the preparation of this symptom are extremely, extremely good. With the symptom 8 in hand, we started to do the solid phase synthesis. We have the resin, we start adding the first symptom, we cleave afterwards the FMOG group, for example, with piperidine, we activate the acid, and we add the next symptom. And so on. We can do this 4 times. We cleave it from the resin and add again this NCS handle at the end so that it can be easily coupled to antibodies. We have now an analog of our DFO star, but this time, as you can see on this table, we have something which is extremely water soluble. Amazingly, I was myself surprised, those 4 atoms of oxygen make the compound completely water soluble. Okay, it's soluble in water like something that we were not believing before. This is fantastic. We wanted afterwards to do radio labeling to demonstrate actually that the oxygen atoms are not playing a role in the binding with the zirconium. We could actually use similar reaction condition than what is used for DFO star and DFO. We could really demonstrate that it was quick to get a good radio labeling yields, radiochemical yields. And We have also demonstrated that DFO, oxo-DFO star is again very stable when we are using challenge experiment. We add again, I think it was 300 times more of a ligand like DTPA and see that the complex is not unstable. And this is exactly what we have observed, contrary to DFO, which is not stable. Now, what do we want? I'm Swiss. We wanted really to increase the opportunity offered by this ligand. And we wanted actually to have as many different biomolecules to be attached to a DFO star derivatives. We had, for example, shown that MCTS is working well to attach to a lysine present in the antibody. But we have also decided to use those type of Michael acceptor so that you can react with a cysteine. Usually malanyamide are used. However, malanyamide, the compound which are the product of the reaction between a malanyamide and a cysteine are usually not very stable in vivo. While the group of Bernardes in Cambridge has demonstrated that, on the contrary, such Michael acceptors are giving compounds which are much more stable. So we have prepared now this analog. We could also couple it to cysteine, and we were very, very happy to have another type of DFO star compound now available. You could see, for example, how beautiful and how clean the compound. Joe is an English postdoc that he prepared in the group, how stable and how nice those compounds are. I'm very happy to say that now the company is selling 3 or 4 different derivatives, the malanyamide, the NCS, just the NH2, and hopefully soon the Michael acceptor. I hope I have convinced you that this compound is extremely interesting. In 2019, the group in Amsterdam has contacted all groups working on this topic in the world and asked them to send their best chelator so that they can make a huge study where they compare each of the chelator to see which one is the best one. And in October 2019, they told us that DFO star was, in their opinion, I have to say, in their opinion, the best compound of what they have received. And in October 2019, they also demonstrating that DFO star was working on rhesus monkeys. So it really worked well also in nearly on men. And in 2019, they have decided now to talk with clinicians in order to have this compound given to patients, hopefully soon. But with the lockdown situation, it has a bit slowed down the problems. I hope I have shown you that DFO star is potentially, we hope, the successor of DFO for imaging with zirconium-89. And I will say that for the moment, it's a topic that we are not working much more any longer because we believe that we have done the chelator which was required. And we hope to see it soon in clinical trial. I want to switch gears and to talk about a fully different topic that our group has been working over the last, I would say, 6-7 years. It is about the use of metal complexes in photodynamic therapy. So PDT is for photodynamic therapy. And I thank all the students which have been working on this topic over the years. Photodynamic therapy is a medical technique which is approved. I really insist on this. It is an approved technique in order to treat certain types of cancer or infection. The idea, you inject intravenously a photosensitizer, you wait until the photosensitizer is accumulating into the tumor, and afterwards you shine light in order to have this photosensitizer reaching first a singlet excited state, and afterwards through intersystem crossing, you will have a reaction between the oxygen, the triplet oxygen present in the tissue, and the photosensitizer to create singlet oxygen, which is extremely reactive, and this reaction will lead to cell toxicity. As you can see, this technique now is used to treat certain types of localized, I insist on these types of cancer, but also of different types of infection. What is very interesting is to say that such compounds which are used for the moment in the clinic, I think there are about 13 compounds which are approved. They have all normally a poor water solubility. The compounds are difficult to synthesize. And very importantly, the compounds are usually accumulating in the skin of the patient, avoiding them to go to the sun for a long time. Why it is that is because normally the majority of the compounds which are on the market are based on porphyrin of phthalocyanine. That's why a few years ago, the groups of Tumour, Glaser, McFarlane, Chow, Mao, Ruiz, Weinstein, and Bryant, and ours, for example, have decided to use metal complexes instead of the usual porphyrins. And very importantly, the group of Sherry McFarlane discovered this TLD1433, which is a very simple ruthenium polypyridine complexes, which is currently in phase 2 clinical trial against bladder cancer. And if everything goes right, it will be probably approved over the next 1 to 2 years. There won't be a phase 3 clinical trial because they have decided to go for types of cancer which cannot be cured for the moment. It is working extremely well, this compound. And it is really a hope for all the medicinal inorganic chemistry community to see such a compound going to clinical trial. I want to discuss a project that Johannes Karges, he was a PhD student in my group until November last year. Now he's a postdoc in San Diego in the group of Sesh Cohen. And it is really a nice collaboration with my colleague at Sun Yat-sen University, Professor Hui Chao. Johannes went to China during his PhD to work with him. So our idea of this project is we wanted really to have ruthenium complexes which can absorb towards the near IR or towards the red. Sorry, I should say first, because the majority of ruthenium complexes, including, for example, TLD1433, are absorbing at about 520, 550 nanometer. But we would like to go even further so that we can treat deep-seated tumors or even large tumors. What we have seen in the literature is that compounds like this, which were developed, I think, in Lyon and in Rennes, have really an absorption which is suddenly going much further towards the red or near IR. And our idea was really to prepare different types of compounds of this type and to assess afterwards the photobiological activity. So Johannes prepared those 6 compounds. As you can see, he has put amine or ester at the end of the polypyridine, polypyridine ligands. And what he could directly see is that the compounds were easy to make. We want always to go for something easy to make in order to go for clinical trial, to go towards the clinic. And you directly see that the compound can be done in one-step synthesis with relatively good yields. He got, for example, the X-ray structure that was Professor Olivier Black who did the crystal structure. Very importantly, afterwards, he did the ruthenium complexes, and in one two-step synthesis, he obtained all the 7 complexes. He even got some X-ray crystal structure, and very importantly, he could see indeed that the compounds that he has prepared had an absorption now up to 650, even maybe 700 nanometer. And this was through a rational design. I'm not talking about this, but we have done also all the TD-DFT calculation before, because we really wanted to demonstrate that we can make a rational design of the compound. Very importantly, and one of the drawbacks of the compound currently in clinical trial, or not in clinical trial, in the clinic, is that they are photobleaching. Once you shine light, they are destroyed. This is the case, for example, of protoporphyrin 9. You can see that as soon as you start shining light, oxygen, singlet oxygen is produced, but you can also see that the photosensitizer itself is destroyed, which is not at all the case with our compound. We have done this with all our compounds, and nearly all of them were extremely stable. We started also to do microscopy, so that all Johannes, who has done this, and he could see that the ruthenium, which is in red here, because they are phosphorescent, there is a triplet state. They could really see that the compounds were accumulating in the cytoplasm. They were not going to the nucleus, they were not going to different organelles. He could also demonstrate this using ICP-MS. And here it is really the beauty to use metal complexes, because you have a metal which is ubiquitous in cells. You can extract each organelle and see how much of the metal is present in these organelles. If you were using, for example, just an organic compound, this would not be possible. So indeed, it was going into the cytoplasm. What we have done afterwards is to demonstrate that the compound were able to produce singlet oxygen in cells. We have made all the quantum years before, but here what you can see, we are using this dye, which is an ester, so that this compound can go into the cells. Once into the cells, because of the presence of esterase, you form now the phenol derivative, which can react with singlet oxygen to make a compound which is fluorescent. Now, I hope it is working, the video. Yes, so as soon as we shine light, you see suddenly the cells becoming bright green because of the fluorescence of DCF. And once again, it is fitting with the localization of the ruthenium. You see the green really outside the nucleus, how I have shown you before. We can also see that the compounds are extremely phototoxic. So these are cervical cancer cells. And what we have done, we have given the compound all in the dark, start shining light, and you see suddenly that the compound is literally bursting through the effect of singlet oxygen production. This is very interesting. This is a mixture of apoptosis and paraptosis. We have also done much more biology because we wanted to show that the compound can be useful. And what we have done, that's Johannes always, he has done little tumors. It's called spheroids. They are between 200 micrometer to 1 millimeter, depending on which technique you are using to grow them. And what you can really interestingly see is that all of the spheroids, so this is now like a sphere, as I will show you here, as soon as, oops. Yes, you can see now, when we go through the Z-axis, the whole sphere, even in the center, is red, indicating that the photosensibilizer is also reaching the middle of the tumor, which is important because quite often metal complexes or even organic drugs are just staying on the crust of the tumor, not killing the core of the tumor. So this is really going to the center. We have afterwards demonstrated that those mini tumors can be killed. You can see that after 3 days, you see that the tumor is growing, growing, growing. We shine light one time after 3 days, and you can see directly that the tumor is decreasing in size. You can see, for example, that compound 7 was the best one here. And this is working at 500 nanometer, but also at 800 nanometer, thanks to 2-photon activation. And this is of high interest because you can go even much deeper at 800 nanometer. Now we wanted also to go even further. We went for mice experiment. You can see here that we are using a mouse. We have given the compound, and what we have found out is that it was accumulating into the intestine very quickly. That is why we have decided to use a cancer model which was related to intestine. We don't know actually why there was an activity into the intestine. This is a healthy, this is a mouse like this. And we have afterwards looked if the compound was toxic to see the toxicity of the compound, you just look at the mass, at the weight, sorry, of the mice over time. If they don't lose weight, it means they are fine, or it's one of the characteristics that you have to look to be sure that mice are not suffering. Usually an animal which is suffering is not eating. And that's why you were very happy to see that the weight of the mice was constant. What we've seen afterwards, it is a single irradiation, either at 500 nanometer, either at 800 nanometer. So one photon, 500, two photon, 800 nanometer. And you can see that we are compound. We have a huge decrease in tumor size, or even really totally an eradication of the tumor. While if we give just water or just the light, the tumor is growing. And this is a very nasty type of tumors that we have grow on the size of the mice. It is a tumor which is resistant to doxorubicin and other types of cancer agent. So we were extremely happy it was working. If you are not convinced, you can look at those pictures that indeed, if you just give compound 7, just make the 1 photon or 2 photon irradiation, you have a big tumor. However, if you give the combination of ruthenium and light, you have a significant reduction of the tumor. I hope I have shown you that now ruthenium complexes have definitively a huge power in PDT, and I really hope that over the next 2 years, the compound of Cherry McFarlane will be in the clinic, and I hope afterwards others will be able to be used, for example, to treat other conditions than bladder cancer. I want to conclude with a project that we have done with my good colleague at Chimie Paritech, PSL University. It's Professor Christophe Thomas. And we have a PhD student together. It's Nancy Soliman. And I thanks also Luc Mackenzie, which is a postdoc in my group in collaboration with Marcel Ollenscheid at the Pasteur Institute. And what we wanted actually is to find a technique to deliver our ruthenium complexes even better to the cells. This story started actually in Zurich. Christina Marie was a PhD student in 2004 in Zurich, and she prepared these simple ruthenium complexes. And she found out that ruthenium complexes had properties, photophysical properties, which was fantastic. It was producing singlet oxygen nearly quantitatively upon light irradiation. This was fantastic. However, when we have done cell testing, no cells were dying. The cells were not at all affected by this compound. This was very disappointing because it was working perfectly in test tube, but not at all when cells. What we have found out afterwards, and that the reason why this compound was not active, it is because it was not at all going into the cells. If it cannot go into the cells, it is impossible to kill cancer cells upon light irradiation. That is why we wanted to have a delivery technique to put this compound in cells. And my colleague, Professor Christophe Thomas, told me we could use actually a polymer, to grow a polymer from the ruthenium in order to have something which can wrap around the ruthenium. What is very important to mention here is that the idea of Christophe Thomas was not to use a typical polymer that you can buy, for example, from Sigma or other companies, but really to grow it covalently from the hydroxyl function present in the photosensitizer. You grow it using lactic acid. So you are doing a polymerization. You will have afterwards the polymer directly attached to the ruthenium, and you can precipitate to form nanoparticles. I just would like to remind you that the lactic can be D or L, and if you use a mixture, you will have an atactic polymer, and if you just use a L or D, you will have an isotactic polymer. What she has done is prepare first the ruthenium, and afterwards using a catalyst developed in the group of Professor Thomas, she could even really grow the polymer in a very, very specific manner. We could really choose
Professor Gilles, you have 5 more minutes.
Yeah, no worries.
5 more minutes.
She could really grow the polymer in a very specific manner to have exactly the number of lactic acid attached to the ruthenium. Here you can see exactly the NMR that Nancy got, and it is really a beautiful NMR. This is just the ruthenium complex, and down it is actually the ruthenium attached to the polymer. And you see really a shift of the CH2 here, the F, which is shifting really significantly upon polymerization. We could do MALDI to prove actually that the compound was formed. You see actually a lot of peaks which correspond to lactic acid. With the compounds in hand, you know, she could make several different lengths of the compound, but she could also make different size of the nanoparticle, depending how you precipitate the compound. And she got afterwards nanoparticle of different size. And it was to observe if the size of the nanoparticle had an effect on the cellular uptake of the ruthenium complex. As you can see here, contrary that if we would have used normal polymers by having this covalent bond, we don't have this burst release directly. It takes a long time to have the ruthenium release from the nanoparticle, exactly how we wanted this. And you can see exactly the proof of our concept on this slide. You see that the ruthenium, RuOH, that Christina prepared in Zurich, you only see a green in the nucleus. The green is actually a dye, a commercial dye. However, there is no purple, which is our ruthenium complex. While using the same concentration, there is the same concentration of ruthenium everywhere on each of those sides. You see that the nanoparticle 4, 3, 2, and 1 hits suddenly the ruthenium in the cell, clearly demonstrating that our concept is working. If you are not convinced with confocal microscopy, you can see with ICP-MS, after 48 hours, we have an important amount of ruthenium now in cells, contrary of the ruthenium by itself. And obviously this correlates very well with the IC50 values. Now our nanoparticles are able to kill upon light irradiation the cells, contrary to what is not possible with the ruthenium. So we have now really a technique in order to bring a compound to cancer cells. So I hope I have shown you that this is working quite well. We are currently using this technique for other purposes, and I would like really to thank Christophe Thomas and Nancy. I would like just to finish, I had prepare something else, but I don't have time. I just want to go to the acknowledgement because I'm very, very lucky to have a lot of dedicated students. Sorry, just taking a bit of time. Tick, tick. Sorry, that's done. I just want really to thank those crowd of people. They're absolutely fantastic. I'm extremely lucky to have those people. They are coming really from all over the world. I would like also to thank the previous students in Zurich. They have done a fantastic job. And all the people we are working with, thanks to them, we are very, very lucky to bring a project much further than we have done by ourselves. Thanks also to the people who have given us money. And thanks again to the organizer for giving me this opportunity. I think I'm right in the 5 minutes. Thank you.
Thank you very much, Professor Gilles, for your wonderful talk on metal complexes in medicinal chemistry. You have highlighted the importance of metal complexes in medicine, though organic compounds are dominating in the pharmaceutical field. After we talk, I think so most of the participants got to know the importance of metal complexes in the medicinal field. And also, I congratulate some of your some of your compounds entered in clinical trials. Good luck to get into trial. Thank you, Professor Gilles, for your wonderful talk. I have here a few participants asked some of the questions led to your talk. Now, because there are many questions, just I group into the questions and I go to the important questions. Remaining one, Loganathan will communicate to you. Okay, then you answer later. The first question we asked, so what are the possible side effects and mode of elimination of such complexes? So, one of the participants.
So they talk about which project?
I think it's related to your like PET.
So for PDT.
Yeah.
Yes, so the side effect, obviously, it is one of the trouble. What is very nice for the moment with the bladder cancer, that's what Sherry McFarlane is doing, is that there should not be any side effect, and the patient have demonstrated so far, to the best of my knowledge, no side effect for a simple and good reason is because the photosensitizer is directly injected within the bladder because it's reachable through the urethra. And because they put only the ruthenium in the urethra, there should not be any side effect because of the complex itself. Now, if you inject by itself the ruthenium, for example, in the vein, it will go a bit everywhere, which can potentially lead to side effect. And this is why we are developing with my colleague, Professor Tumour, manner to deliver the ruthenium in the best possible selective manner to the tumor to lower the amount of complex to the place where you want. I just give an example. It seems that only 1 to 2% of platinum, which is given to a patient during a chemotherapeutic treatment, which is actually going to the nucleus, to the DNA of the cell. 98% is going somewhere where it should not go, leading to the severe side effect, which is observed with cisplatin, but still it is used every day. So definitely it's something that we are aware. And all I can say for the moment, it's on mice, we didn't observe any side effect. What we have observed is no social problems. They were not fighting each other. They were not going to a side of the cage, and they did not stop eating. That's what we can say for the moment, but this is really an important question that cannot be answered as long as we did not do more experiments. But thanks a lot. That's good.
Another question from one of the participants. Can we use this metal complex as a dual purpose as for imaging and therapeutic? If yes, how? Can we use it as a theranostic agent like that?
Yeah, it is also a good question. Yes, obviously the compound is phosphorescent. Okay, so you can see it. The only trouble for the moment is fluorescence imaging is not ideal because you cannot go very, very deep. You cannot see phosphorescence very deep. It's working quite well, for example, with mice. However, it is much more problematic, in human. However, what I strongly believe, the hope, it is for fluorescence guided surgery. When you give the photosensitizer and afterwards, since you anyway, operate the patient, you have surgery. When you open, you will be able to see the localization of the photosensitizer, and when you will shine light in the body of the patient, you will see where exactly is the tumor. It is already in the clinic. It is already approved, for example, for brain cancer. So you give the photosensitizer, and you have this dual effect between imaging and therapy thanks to PDT. So yes, the students or the professor is absolutely right by asking this.
Another question, sir. Is there any criteria used in selecting metal for any specific activity?
Oh, can you just repeat? I did not catch.
Is there any criteria used in selecting metal for any specific activity? So what are the criteria in choosing the metal complex for a particular disease? The question is.
Oh, that's a good question. So technically, is there a metal which is really specific? To the best of my knowledge, for example, like I have done with the zirconium, it's not the metal giving the specificity. It is the antibody which is enabling to have the specificity. However, is there a metal which is really specific? Well, one can actually argue that the ruthenium complexes from Professor Kepler, which has ruthenium-3, which has reduced to ruthenium-2 and becoming active because under hypoxia, because tumors have a reducing environment, it is kind of a selectivity. So yes, I could say metals can be selective if, for example, you are using those properties of redox state, reduction. It's called the famous activation by reduction, which was discovered with the NAMI-A and afterwards with Kepler's compound. So yes, it is possible.
Another question related to PDT. How is PDT used to treat tumors in the brain?
In the brain? Ah, okay.
Yeah,
that's
the brain. Tumors in the brain.
That's a very good question, and it is actually used. Technically, all tumors normally in the brains required surgery. And what you do is that you inject the compound, and afterwards you will open the skull. This is what is currently done for people having this terrible disease, brain cancer. And when you open, when you debulk, you remove the tumor, you will be able to use light. So there is really a teamwork between the surgeon and the specialist of PDT, which can be sometimes the same. So there is really the use of surgery and PDT, which is possible. However, it is also possible sometimes to avoid surgery, like in the case of bladder cancer or prostate cancer, okay? But in the case of brain, it is working, and it is used, for example, in Germany, in Groningen or in München. Yes?
The last question from my side. So what's the penetration capacity of these ruthenium complexes? Because how deeper we can go on imaging or work for PDT. Is the PDT, we can't go for, now people are saying more than 10 mm. So in that case, how far we can go?
Okay,
with this
is also a very good question. Technically, we say that if you go to 650 nanometer or higher, you can go up to 2 centimeter. Okay, the light can go to 2 centimeter, and if the photosensitizer is 2 centimeter lower, you will be able to destroy the tumor. But once again, I would like really to remind people, PDT is not an alternative. It is a complementary technique, okay? Surgery, chemotherapy will always be used. However, you can increase the chance of survival of the patient by combining. We all know this, and that's why, I'm always writing in our paper, it is a complementary technique, and I will encourage people not to believe that it is an alternative. It can sometimes, but I think it is in addition to, okay? So you will debulk, you will remove the tumor, but for example, in the case of brain, you cannot debulk because, the brain is important. Maybe afterwards the patient won't be able to walk. That's why you can just take what is necessary. So you take first with the scalpel, but afterwards you finish up the work with light. Okay, I hope it's replied to your question.
Thank you, Professor Gilles. Due to lack of time, so I can't read all the questions. There are many questions.
My apology to the students.
Yeah. No, Dr. Loganathan may forward those questions to you. You can answer later. Thank you very much, Professor Gilles, for our excellent talk and answering the questions from the participants. Thank you very much, Dr. Loganathan, and the organizing committee from VIT for giving me this wonderful opportunity to chair this session. Thank you very much, one and all.
Yeah,
thanks a lot.
On behalf of organizing committee, Vellore Institute of Technology and Swansea University, UK, I would like to thank Dr. Gilles for his excellent talk on metal complexes in medicinal chemistry. And also, for explaining how DFO star NCS compound developed from your lab helping in immunopet applications. Congratulate Gilles for commercializing DFO star NCS compound. And also for I think it will another compound DFO star NBX2 and maleimide compound will be commercialized soon. We are happy to hear about your recent result on DFO stars in preclinical animal models. I hope it will reach all the regulatory approval and it will reach market. Congratulate and good luck, Gilles. This is really a bench to benchmark research. This particular session, 700 and nearly 800 participants participated in this session. I would like to thank all the participants for actively participating in many of the sessions and also for this particular session. Thank you all. For information for the participants, the next session will start at 7 p.m. Professor Andrea Kaczynski from Purdue University is joining at 7 p.m. Thank you all for your wonderful contributions. Thank you, Gilles, and thank you, Dr. Rajendran, for sharing this session. Thank you all.
Thank you. Take care.
We are closing this session. Thank
Yes,
you.
yes, yes.